Right now, 4 astronauts who just flew around the moon are being studied for damage they cannot feel. Artemis 2 splashed down in April 2026, and NASA is quietly scanning the crew for changes that started the moment they left Earth's magnetic shield. Not burns, not broken bones, something worse. Their bones may already be thinner, their eyes may already be reshaping, their DNA may already carry mutations that will not show symptoms for years. Deep space does not kill you all at once. It disassembles you piece by piece, organ by organ, cell by cell, so slowly that you would not know it was happening until it was too late. What you are about to hear is the most unsettling truth in human spaceflight. If you find this kind of deep science storytelling valuable, consider hitting that like button and subscribing. It genuinely helps this channel keep making content like this, and it means more than you might think. Now, settle in. Get yourself comfortable. Let's begin. Ask anyone what scares them about deep space and they will describe something violent. A hull breach that sucks the air from the cabin in seconds. A micrometeorite puncturing a fuel line. An engine failure stranding a crew millions of miles from home with no hope of rescue. A fire erupting inside a sealed capsule where there is nowhere to run. These are the nightmares we carry because they are visible, dramatic, and easy to imagine. Hollywood has spent decades reinforcing them. Explosive decompression, freezing vacuum, the silent scream of an astronaut tumbling into the dark. And none of it is wrong. These catastrophic scenarios are real. They have killed cosmonauts and astronauts alike. The crew of Soyuz 11 died in 1971 when a ventilation valve opened during capsule separation and depressurized their spacecraft in the vacuum of space. 3 of them suffocated before the spacecraft reached the ground. The crew of Apollo 13 nearly froze to death when an oxygen tank ruptured halfway to the moon and crippled the command module. The Space Shuttle Challenger disintegrated 73 seconds after launch. Columbia broke apart during re-entry. These are not theoretical fears. They are entries in the historical record, written in human lives. But here is what almost nobody talks about. These disasters, as horrifying as they are, represent the kind of danger we understand. Mechanical failure, structural compromise, sudden catastrophic loss. The body is destroyed in an instant, and the cause is obvious. There is a rupture. There is a fire. There is a failure. The threat is visible. The death is immediate. The investigation finds the broken part. The real horror of deep space is nothing like this. The real horror is what happens when everything works perfectly. When the spacecraft holds. When the seals remain tight. When the air stays breathable, the water stays clean, the food supply holds, and every system on board performs exactly as designed. Because even inside a flawless spacecraft, even with every piece of engineering functioning at peak performance, the human body begins to come apart. Not suddenly, not dramatically, not in any way that would trigger an alarm or alert mission control. It happens in silence. It happens in the bones and the blood and the fluid behind the eyes. It happens in the cells and the marrow and the architecture of the brain. It begins within hours of leaving Earth, and it does not stop. It accelerates, it compounds, and most of the damage cannot be felt by the person it is destroying. This is the invisible threat that defines the real challenge of deep space exploration. Not a broken spacecraft. Not a failed engine. A broken body still breathing, still conscious, still performing tasks and running checklists, while its internal systems are being slowly and silently dismantled by an environment the human organism was never designed to survive. To understand why this happens, you first have to understand what Earth does for you without your knowledge. Every second of every day, your body is engaged in a constant negotiation with gravity. Your skeleton bears weight. Your muscles resist downward pull. Your cardiovascular system pumps blood against a vertical pressure gradient that stretches from your feet to your skull. Your inner ear detects gravitational orientation and feeds that information to your brain, which uses it to maintain balance, posture, and spatial awareness. Your bones are not static structures, they are living tissue under constant renovation. Specialized cells called osteoblasts build new bone, while another type called osteoclasts break old bone down. On Earth, This cycle is balanced by mechanical loading, the force that gravity and physical activity impose on your skeleton every time you stand, walk, or lift something. Your body senses that load and responds by maintaining bone density in the regions that bear the most weight. This entire system evolved over hundreds of millions of years under one specific condition: a gravitational pull of 9.8 meters per second squared at the surface of a rocky planet with a dense iron core. That is not a variable your biology can negotiate. It is the foundational assumption on which every structural and circulatory system in your body is built. The moment you leave Earth and enter microgravity, that assumption collapses, and everything built on top of it starts to fail. Bone loss in microgravity is one of the most thoroughly documented effects of spaceflight, and it is also one of the most quietly alarming. NASA and the European Space Agency have tracked bone mineral density in astronauts for decades. The data is consistent and disturbing. In the absence of gravitational loading, astronauts lose roughly 1 to 2% of bone mineral density per month in weight-bearing bones. The hips, the spine, the legs. That rate was first observed during the Skylab missions in the mid-1970s and has been confirmed repeatedly in long-duration crews aboard the International Space Station. To put that in perspective, a postmenopausal woman on Earth, the population most vulnerable to osteoporosis, loses roughly 1 to 1.5% of bone density per year. An astronaut in microgravity matches that annual loss in a single month. A 6-month mission aboard the station can produce bone loss equivalent to what an aging person on Earth would experience across an entire decade. And the loss is not uniform. Non-weight-bearing bones, like those in the skull or the forearms, are mostly spared. The skeleton is not dissolving evenly. It is being selectively eroded in the places that matter most for standing, walking, and absorbing impact, precisely the structures you would need to function on a planetary surface after landing. The femoral neck, the lumbar vertebrae, the tibial cortex. These are the bones that fracture most catastrophically in elderly patients on Earth. They are the same bones that microgravity attacks first. What drives this loss is a disruption in the remodeling cycle. Without mechanical loading to signal that bone density is needed, the body downregulates osteoblast activity, the cells that build new bone, while osteoclast activity continues or even increases. Bone resorption begins outpacing bone formation within the first 2 weeks of spaceflight. Urinary markers of bone breakdown spike almost immediately. The calcium liberated from dissolving bone floods the bloodstream and has to be filtered by the kidneys, which raises the risk of kidney stones, a secondary complication that is dangerous on its own in an environment where surgical intervention is not available. That single sentence deserves much more attention, because the kidney stone risk is not an abstract statistical footnote. It is a concrete medical emergency that connects bone loss to organ damage through a chain of events that most people would never think to link. Here is how it works. When osteoclasts dissolve bone tissue in microgravity, they release calcium and phosphate directly into the bloodstream. On Earth, this released calcium would be partially reabsorbed by new bone being built elsewhere in the skeleton. But in microgravity, where bone formation is suppressed and bone resorption is elevated, the calcium has nowhere to go. It circulates. The kidneys must filter it. Urinary calcium concentrations rise sharply. Studies of astronauts aboard the station have documented increases of 40 to 60% in urinary calcium excretion during the first weeks of spaceflight. This elevated calcium saturates the urine and combines with oxalate and phosphate ions to form crystalline deposits. The technical term is nephrolithiasis. The common term is kidney stones. On Earth, kidney stones are excruciatingly painful but rarely life-threatening. The patient goes to an emergency room, receives pain management and hydration, and in most cases passes the stone naturally or has it broken apart with focused ultrasound. The entire episode resolves within days. On a spacecraft in deep space, none of that infrastructure exists. There is no emergency room. There is no ultrasound lithotripsy device. The onboard medical kit includes analgesics and a limited supply of fluids, but if a stone lodges in the ureter and obstructs urine flow, the crew faces a surgical emergency with no surgeon and no operating theatre. An obstructed kidney can become infected within days, progressing to sepsis in a patient whose immune system, as we will soon discover, is already compromised by radiation and microgravity. A kidney stone that would be a painful inconvenience on Earth becomes a potentially fatal event in deep space. And the risk is not theoretical. At least one astronaut aboard the station has been documented passing a kidney stone during a mission, and post-flight imaging has revealed asymptomatic stones in others, stones that formed silently during the mission and were discovered only after return to Earth. The countermeasure currently used on the station is aggressive hydration and dietary modification to reduce urinary calcium concentration, but compliance is difficult in an environment where water is a managed resource, and the sensation of thirst is suppressed by the fluid redistribution that sends blood and fluid toward the head. The calcium is not the only metabolic burden the kidneys must manage. As we will see, microgravity also accelerates the destruction of red blood cells, releasing iron and hemoglobin degradation products into the bloodstream that must be processed and excreted. The kidneys become a bottleneck for multiple waste streams simultaneously: calcium from dissolving bones, iron from destroyed blood cells, and the normal metabolic byproducts of daily cellular function, all while operating inside a body whose total blood volume has been reduced by 10 to 15%, and whose cardiovascular system is delivering less perfusion, pressure than it would on Earth. The organ responsible for filtering the blood is being asked to handle a heavier workload with a smaller supply for months or years without relief. This cascade from silent bone loss to elevated blood calcium to saturated urine to crystalline deposits to potential obstruction illustrates a principle that will appear again and again throughout this story. The damage in deep space is not confined to the system where it originates. It migrates. Bone loss becomes a kidney problem. A kidney problem becomes an infection risk. An infection risk becomes a survival question. Each system's failure feeds downstream into systems that appear unrelated until you trace the biochemistry connecting them. Astronauts aboard the station exercise for roughly 2 hours every day using a device called the Advanced Resistive Exercise Device, which simulates the loading effects of weight training by using vacuum cylinders to create resistance. This countermeasure helps. It reduces the rate of bone loss compared to crews without access to it, but it does not eliminate the loss. Even with aggressive daily exercise, astronauts still return from 6-month missions with measurable bone deficits, and a study published in 2022 found that some of that lost bone architecture, particularly trabecular microstructure in the tibia, had not fully recovered even 1 year after return to Earth. For missions longer than 12 months, the question becomes whether certain structural losses cross a threshold beyond which full recovery is no longer possible. This is the detail that transforms bone loss from a manageable inconvenience into a deep space crisis. On the station, missions last 6 months to a year, and the crew returns to full Earth gravity and medical support. Recovery is slow but generally achievable, but a Mars mission involves approximately 900 days from launch to return, with up to 300 days of deep space transit in each direction, and roughly 500 days on a planetary surface with only 38% of Earth's gravity. The bones that erode during transit do not rebuild during the surface stay. Mars gravity is too weak to fully reload the skeleton. By the time the crew boards the return vehicle, they may have spent 2.5 years in conditions that never stop draining their bone density. The math becomes difficult to escape. At 1.5% loss per month in microgravity, a 9-month outbound transit alone could reduce hip bone density by over 13%. Combine that with incomplete recovery on Mars and another 9 months of transit home, and the crew may arrive back on Earth with skeletal systems that resemble those of 70 or 80-year-old patients regardless of their actual age. Muscle atrophy follows a parallel trajectory, and in some ways it is even faster. Anti-gravity muscles, the soleus in the calf, the quadriceps, the paraspinal muscles that hold the spine upright, begin wasting within days of entering microgravity. Without the constant demand of resisting gravity, the body treats these muscles as metabolically expensive tissue that is no longer needed. Protein synthesis decreases, protein degradation increases. Muscle fibers shrink in cross-sectional area and shift from slow-twitch endurance fibers to fast-twitch types that fatigue more quickly. Studies of astronauts returning from 6-month station stays have documented calf muscle volume losses of up to 20% and strength reductions of up to 40% in specific muscle groups, even with rigorous daily exercise protocols. The exercise countermeasures help with muscle preservation more than they help with bone. Resistance training aboard the station has significantly reduced the severity of muscle loss compared to earlier space station eras, but the protection is incomplete. Muscles that are difficult to isolate with machine-based exercise deep spinal stabilizers, small postural muscles in the feet and ankles continue to atrophy despite the crew's best efforts. And the neurological component of muscle function degrades too. The brain's motor programs for coordinating movement under gravity grow stale from disuse. Returning astronauts frequently report balance problems, coordination deficits, and difficulty walking in the first days and weeks after landing. Most recover within a few months, but the recovery depends on full gravitational reloading and access to rehabilitation conditions that would not exist on Mars. There is another musculoskeletal change that almost no one outside of aerospace medicine talks about, and it reveals how deeply microgravity reshapes the body's architecture in ways that are both invisible and misleading. In the absence of gravitational compression, the spinal column elongates. The intervertebral discs, the cartilage pads that sit between each vertebra, acting as shock absorbers, and flexible joints expand as the compressive load that normally squeezes them all day on Earth disappears. Without that load, the discs absorb fluid from the surrounding tissue and swell. The cumulative effect across the entire spine is significant. Astronauts routinely grow taller in microgravity by approximately 3 to 5 centimeters, roughly 1.5 to 2 inches. Some crew members have reported height gains at the upper end of that range within the first week. This sounds like a curiosity. A fun fact for trivia night. It is actually a clinical problem. The expansion of the discs stretches the ligaments and small muscles that stabilize the spinal column. This produces lower back pain in approximately half of all astronauts during long-duration missions, a rate significantly higher than in the general population on Earth over the same time period. The pain is typically described as a deep, diffuse ache in the lumbar region that is difficult to localize and does not respond well to the stretching or positioning adjustments that would relieve it on the ground. In microgravity, there is no posture that fully unloads the spine because there is no gravitational vector to unload against. The discs remain expanded continuously, and the surrounding tissues remain stretched continuously for the entire duration of the mission. But the more serious problem is not the pain itself. It is what the expanded discs become when gravity returns. A disc that has been swollen with fluid for 6 months is structurally different from a disc that has been under normal compressive load. The collagen fibers in the annulus fibrosus, the tough outer ring that contains the gel-like nucleus of the disc, have been stretched beyond their normal operating range. When the astronaut returns to Earth and gravitational compression suddenly resumes, these compromised discs must absorb impact forces they are no longer well configured to handle. The risk of disc herniation, where the nucleus pulposus bulges or ruptures through the weakened annulus, is significantly elevated in the post-flight period. Research published by NASA-affiliated investigators found that astronauts had approximately 4 times the incidence of cervical and lumbar disc herniation compared to matched controls, with the herniations occurring predominantly in the first year after return from long-duration missions. A herniated disc can compress spinal nerves, producing pain, numbness, weakness in the extremities, and in severe cases, loss of bladder or bowel control. On Earth, surgical intervention is available. In deep space or on Mars, it is not. And the insidious quality of this risk matches the pattern of every other form of damage described in this script. The astronaut feels back pain during the mission, assumes it is muscular, adapts to it, and continues functioning. The underlying disc vulnerability is structural, progressive, and invisible on routine examination. It reveals itself only when load returns, when the crew lands, when someone bends to lift equipment on a planetary surface, when the spine absorbs a force the compromised disc cannot distribute. The spinal changes also interact with the bone loss already described. The vertebral bodies themselves are among the bones most affected by microgravity-induced mineral loss. A weakened vertebra supporting a swollen, structurally compromised disc creates a compound vulnerability. The load-bearing surface and the load-distributing cushion are both degraded simultaneously. On Earth, this combination is seen almost exclusively in elderly patients with advanced osteoporosis. In space, it can develop in a healthy 35-year-old over the course of a single mission. The interplay between bone and muscle loss creates a compounding problem that is worse than either system alone. Bones depend on muscle loading to maintain density. Muscles depend on skeletal stability to generate force. As muscles atrophy, they transmit less mechanical force to the bones they attach to, accelerating bone loss. As bones weaken, the joints they support become less stable, increasing the risk of injury during the very exercise that is supposed to protect both systems. The body enters a feedback loop of structural decline, where each system's deterioration worsens the other. And all of this unfolds beneath the astronaut's conscious awareness. You cannot feel your bones thinning. You cannot feel your muscle fibers shrinking at the cellular level. You can feel weakness if it becomes severe, but in the reduced demand environment of a spacecraft where you float rather than walk, push off walls rather than climb stairs, and never lift anything heavy because nothing has weight, the functional consequences of muscle loss are masked. The body feels adequate for the tasks at hand. The damage reveals itself only when the demands change, when gravity returns, when the crew has to walk on a planetary surface, when someone stumbles and a weakened femur absorbs an impact it can no longer withstand. But the musculoskeletal system is only the most intuitive layer of the damage. Beneath it, something stranger and more insidious is happening. A redistribution of the body's internal fluids that will ripple outward into the eyes, the brain, and the cardiovascular system in ways that researchers are still working to fully understand. On Earth, gravity pulls your blood and body fluids downward. Roughly 70% of your blood volume resides below your heart at any given moment when you are standing. Your cardiovascular system is designed around this distribution. Your heart pumps against this gradient. Your leg veins contain one-way valves that prevent blood from pooling. Your baroreceptors, pressure sensors in the carotid arteries and aortic arch, constantly monitor this distribution and adjust heart rate and blood vessel tone to maintain stable blood pressure regardless of posture. In microgravity, the downward pull vanishes. Without gravity sorting fluids vertically, approximately 1.5 to 2 liters of blood and interstitial fluid shift from the legs toward the torso and head. This is not a subtle change. Astronauts experience it immediately. Their faces swell, their sinuses congest, their legs become visibly thinner. Veteran crew members describe the sensation as feeling like hanging upside down permanently, a pressure in the head that diminishes over days but never fully resolves. The cardiovascular system responds to this redistribution as if the body has suddenly gained too much blood volume. Sensors in the heart and major vessels detect the increased central fluid pressure and trigger a cascade of hormonal adjustments. The kidneys excrete more water and sodium to reduce plasma volume. Total blood volume decreases by roughly 10 to 15% within the first few days. The heart, now receiving less blood per beat and working against less gravitational resistance, begins to remodel. The left ventricle, the chamber that pumps blood to the entire body, loses mass. Cardiac output decreases. The heart becomes physically smaller. This cardiac atrophy is measurable on imaging. It is not dramatic enough to impair function in microgravity, where the heart's workload is reduced anyway. But it represents a structural change to the most critical organ in the body, and it creates a dangerous mismatch upon return to gravity, when the cardiovascular system suddenly has to pump blood uphill again with a heart that has partially remodeled for an environment that no longer exists. Orthostatic intolerance, the inability to maintain blood pressure while standing, affects a significant percentage of returning astronauts. Some cannot stand upright for more than a few minutes without becoming lightheaded or losing consciousness. On Earth, with medical teams standing by, this is manageable. On Mars, after 9 months of transit, it could be mission-threatening. The fluid shift also pressurizes the venous system in the head and neck in ways that have unexpected consequences. Jugular veins, which on Earth drain blood downward from the brain, distend and slow their flow in microgravity. In 2019, researchers discovered that one astronaut aboard the station had developed a blood clot in the internal jugular vein, the first confirmed case of deep vein thrombosis in space. Subsequent screening revealed sluggish or stagnant flow in the jugular veins of multiple crew members. The clinical significance of this finding is still being studied, but the implication is clear. Microgravity does not simply redistribute fluids, It creates conditions where the blood itself may behave differently, pooling in regions where it should flow freely, clotting where it should remain liquid. And this fluid redistribution is not a temporary adjustment that resolves once the body acclimates. It persists for the duration of the mission. The body reaches a new equilibrium, a microgravity equilibrium that is fundamentally different from its terrestrial state. The head remains pressurized, the cardiovascular system remains remodeled, the blood volume remains reduced. Every day in microgravity is another day the body spends adapted to an environment that will eventually end, at which point it must rapidly readapt to conditions it has been structurally departing from for months or years. Here is the pattern that makes deep space so disturbing as a biological environment. None of these changes announce themselves. Bone loss produces no pain. Muscle atrophy in microgravity produces no functional impairment until gravity returns. Fluid redistribution causes discomfort but not alarm. Cardiac remodeling is detectable only by imaging. Jugular flow stagnation requires ultrasound to discover. An astronaut experiencing all of these changes simultaneously could feel essentially normal, adequately strong, cognitively sharp, physically comfortable, while their skeleton, their muscles, their heart, and their circulatory system are all quietly drifting away from the configuration that would allow them to survive on any planetary surface. This is what makes the invisible damage so different from the catastrophic dangers most people imagine. A hull breach kills you, and you know it. Bone loss at 1.5% per month erodes your skeleton across weeks and months while you float through your daily routine. Check your email, run your experiments, and sleep soundly. The threat is not an event. It is a gradient. It is a slow curve bending in the wrong direction, hidden behind the body's remarkable ability to compensate for damage it cannot yet feel. And everything described so far, the bones, the muscles, the fluid, the heart, the veins, is only the structural layer. Beneath it, another assault is underway, one that operates at the level of cells and chromosomes, one that the body has even less ability to detect, repair, or resist. And one that gets profoundly worse the moment a spacecraft leaves the protective reach of Earth's magnetic field and enters the full radiation environment of deep space. That is where the damage stops being merely structural and becomes something far more difficult to survive. Everything described so far happens inside the International Space Station, which orbits roughly 400 kilometers above the surface of Earth. That is close. Cosmically speaking, it is barely off the ground. The station circles within the outermost boundary of Earth's magnetosphere, the vast magnetic bubble generated by the churning liquid iron in our planet's core. This bubble extends tens of thousands of kilometers into space and deflects the majority of high-energy charged particles streaming through the solar system. It does not block everything. Astronauts aboard the station receive radiation doses roughly 100 times higher than the average person on the ground. But the magnetosphere absorbs or redirects the most dangerous particles, the ones traveling fast enough and carrying enough energy to shatter molecular bonds inside living cells. Step beyond that shield and the radiation environment changes completely. This is the distinction that separates station medicine from deep space medicine, and it is the distinction that makes long-duration missions beyond Earth orbit a fundamentally different biological problem. On the station, microgravity is the primary assailant. Radiation is a secondary concern, elevated but manageable within NASA's career exposure limits. In deep space, on a transit to Mars, on the lunar surface during a solar storm, anywhere beyond the magnetosphere's protection, radiation moves from the background to the foreground. It becomes a constant, penetrating, and largely unshieldable threat that attacks the body at the molecular level every second of every day for the entire duration of the mission. There are two primary sources of radiation in deep space, and they behave very differently. The first is solar particle events, bursts of energetic protons ejected from the sun during solar flares and coronal mass ejections. These events are episodic and sometimes violent. A major solar particle event can deliver a radiation dose in hours that would normally accumulate over months. The particles are mostly protons, which are relatively light and can be partially blocked by spacecraft shielding, aluminum hull material, water tanks, and polyethylene barriers. Solar events are dangerous but intermittent, and they can be partially anticipated. Space weather monitoring provides some warning time. Crew can retreat to a more heavily shielded area of the spacecraft, a storm shelter, and wait for the event to pass. The Artemis II mission, which flew its crew around the Moon in April 2026, carried dosimeters and radiation monitoring instruments specifically designed to characterize this environment during solar maximum. When such events are most frequent, the second source is far more insidious. Galactic cosmic rays are not bursts. They are a permanent background, a steady rain of high-energy particles originating from supernova remnants, neutron stars, and other violent astrophysical sources across the Milky Way. Unlike solar particles, which are mostly protons, galactic cosmic rays include fully ionized atomic nuclei of every element on the periodic table, from hydrogen all the way up to iron and beyond. These particles travel at significant fractions of the speed of light and carry enormous kinetic energy. A single iron nucleus in a galactic cosmic ray can carry the energy of a baseball thrown at 60 miles per hour, concentrated into a particle smaller than an atom. This is what makes galactic cosmic radiation fundamentally different from any radiation source on Earth and from the solar radiation that dominates the near-Earth environment. These heavy ions, classified in radiation biology as HZE particles, meaning high atomic number and high energy, do not merely pass through tissue. They carve destructive tracks through it. As an HZE ion travels through the body, it strips electrons from every molecule in its path, generating a dense column of ionization damage called a primary track. But the destruction does not stop there. The electrons torn loose by the primary ion are themselves energetic enough to cause secondary ionizations, creating branching trails of damage called delta rays that extend outward from the main track by distances measured in millimeters. A single ion passing through a cell nucleus can break both strands of the DNA helix in multiple locations simultaneously, a type of damage called a clustered lesion that is extraordinarily difficult for the cell's repair machinery to fix correctly. On Earth, the atmosphere and magnetosphere together absorb virtually all HZE particles before they reach the surface. Humans evolved with zero exposure to this type of radiation. The body's DNA repair mechanisms, which are sophisticated and generally effective against terrestrial radiation sources like ultraviolet light and natural background gamma rays, were never designed to handle clustered damage from heavy ions. The repair enzymes can fix a clean single-strand break in minutes. A double-strand break takes longer and sometimes introduces small errors, but a clustered multi-site lesion caused by an HZE track may overwhelm the repair system entirely, leading to permanent mutations, chromosomal rearrangements, or cell death. The dose rates from galactic cosmic radiation in deep space are not extreme by the standards of, say, a nuclear accident. An astronaut on a Mars transit would accumulate roughly 1.8 millisieverts per day from galactic cosmic rays alone, a rate that translates to approximately 660 millisieverts per year, or roughly 0.66 sieverts. For comparison, a single chest X-ray delivers about 0.02 millisieverts. A year in deep space delivers the equivalent radiation of approximately 33,000 chest X-rays. That sounds alarming, and it is, but the raw dose number alone does not capture what makes galactic cosmic radiation so biologically threatening. The danger lies not in the total energy deposited, but in how that energy is deposited in concentrated, devastating tracks rather than the diffuse scatter of X-rays or gamma rays. The biological effectiveness of HZE ions per unit of dose is estimated to be anywhere from 5 to 20 times greater than that of X-rays for certain types of tissue damage, particularly cancer induction and central nervous system effects. But the central nervous system and the genome are not the only targets. There is a third system that radiation quietly degrades, one that connects the structural damage already described to an entirely new category of long-term risk. The cardiovascular system, already remodeling under the influence of microgravity and fluid redistribution, is simultaneously being attacked by radiation through a completely independent mechanism. On Earth, cardiovascular disease develops over decades through a slow process of arterial damage, inflammation, and plaque accumulation. The inner lining of blood vessels, the endothelium, is a single cell-thick layer that regulates blood flow, controls inflammation, and prevents clotting. When that lining is damaged by high blood pressure, high cholesterol, or chronic inflammation, the body mounts a repair response that, over time, produces the fatty plaques characteristic of atherosclerosis. These plaques narrow arteries, restrict blood flow, and can rupture to trigger heart attacks or strokes. The entire process takes 30 to 50 years under normal conditions, which is why cardiovascular disease is overwhelmingly a disease of middle and old age. Radiation accelerates this timeline dramatically. Studies conducted at NASA's Space Radiation Laboratory using simulated galactic cosmic radiation have demonstrated that exposure to heavy ions produces measurable damage to the endothelial lining of blood vessels in animal models. The radiation does not need to kill endothelial cells outright. It induces oxidative stress within them, triggers inflammatory signaling cascades, and alters the expression of genes that regulate vascular tone and repair. The endothelium shifts from a quiescent, protective state to an activated, pro-inflammatory state. The same transition that initiates atherosclerosis on Earth, except that radiation can trigger it in weeks rather than decades. Researchers have observed that animals exposed to space-relevant doses of heavy ions develop increased arterial stiffness, a loss of elasticity in the vessel walls that forces the heart to work harder with each beat. They develop early-stage atherosclerotic changes in the aorta and carotid arteries. They show elevated markers of vascular inflammation that persist long after the radiation exposure ends. And these effects are distinct from the cardiovascular remodeling caused by microgravity. Fluid redistribution in microgravity causes the heart to shrink and cardiac output to decrease, a structural adaptation to reduced workload. Radiation causes the blood vessels themselves to stiffen and inflame, a form of accelerated vascular aging that increases workload on a heart that is simultaneously atrophying from disuse. These two processes, a shrinking heart and stiffening arteries, are moving in opposite directions. The heart is becoming weaker, while the vessels it pumps blood through are becoming harder to perfuse. On Earth, this mismatch is the hallmark of age-related cardiovascular decline and is the leading cause of heart failure in elderly populations. In deep space, it could develop in an otherwise healthy crew member across a matter of months or years. NASA's Human Research Program now lists cardiovascular degenerative disease as a distinct risk category for deep space missions, separate from cancer. The concern is not that an astronaut will have a heart attack during a Mars transit, though that possibility cannot be excluded, but rather that the radiation-induced vascular damage will compound over the mission duration and manifest as premature cardiovascular disease in the years and decades following return. An astronaut who completes a 3-year deep space mission at age 40 might carry a cardiovascular risk profile more typical of someone 20 years older. And unlike cancer, which can sometimes be detected and treated, early-stage vascular damage is largely asymptomatic. The arteries stiffen, the endothelium inflames, the plaques begin forming, and the astronaut feels nothing because cardiovascular disease, like every other form of invisible damage deep space inflicts, operates below the threshold of perception until the day it produces a clinical event. Think of it as the body's internal plumbing slowly corroding from the inside. The pipes still carry water, the pressure still holds, everything appears functional. But the walls are thinning, the joints are weakening, and the system is aging faster than the calendar says it should. By the time a leak appears, the corrosion has been spreading for years. This means that radiation in deep space is not merely a cancer risk and a cognitive risk. It is a cardiovascular risk that converges with the cardiac atrophy and fluid redistribution caused by microgravity to create a compounding assault on the circulatory system, the system responsible for delivering oxygen and nutrients to every other organ in the body. If the circulatory system degrades, every other system degrades with it. The brain receives less oxygen, the muscles receive less fuel, the kidneys filter less efficiently, the immune cells travel more slowly to sites of infection. The cardiovascular system is not just another item on the list of deep space damage. It is the supply chain that connects everything else. And the assault extends beyond structural and vascular damage to a risk that is measured not in symptoms but in statistics. NASA's current career radiation exposure guidelines are designed to limit the increased risk of exposure-induced death from cancer to a defined threshold. Under those guidelines, a round-trip Mars mission lasting roughly 900 days would bring most astronauts uncomfortably close to or beyond their career limits. And that estimate assumes no major solar particle events during transit. A single large solar storm encountered outside the magnetosphere could push cumulative exposure well past the limit in a matter of hours. But cancer risk, as serious as it is, represents only one dimension of the radiation problem, and it may not be the most operationally dangerous one. Beginning in the mid-2000s, researchers at NASA's Space Radiation Laboratory at Brookhaven National Laboratory began systematically exposing rodent models to simulated galactic cosmic radiation beams of heavy ions, calibrated to mimic the energies and particle types astronauts would encounter in deep space. The results were consistent and deeply troubling. Animals exposed to space-relevant doses showed persistent cognitive impairments that did not resolve over time. In some cases, the deficits worsened months after exposure. A landmark study published in 2023 used a complex 33-beam simulation of galactic cosmic radiation, the most realistic ground-based approximation of the deep space radiation environment ever created, and exposed mice to both acute and chronic dose profiles. The researchers found that galactic cosmic radiation did not alter reward sensitivity, meaning the animals' basic motivational drives remained intact. But it significantly impaired attentional processes and increased reaction times. In the prefrontal cortex, the brain region most critical for executive function, decision-making, and behavioral flexibility, the radiation disrupted dopamine signaling and reorganized the functional connections between neurotransmitter networks. The damage was not simply a matter of killing neurons, it was a rewiring. The architecture of how the prefrontal cortex communicated with itself was altered in ways that persisted long after the exposure ended. Earlier studies at the same facility had demonstrated that space-relevant doses of heavy ions reduced dendritic complexity in hippocampal and cortical neurons. Dendrites are the branching extensions of nerve cells that receive signals from neighboring neurons. They are the physical substrate of neural connectivity. Fewer and simpler dendrites mean fewer synaptic connections, which translates directly into reduced capacity for learning, memory consolidation, and cognitive flexibility. Spine density, the number of tiny protrusions on dendrites where synapses form, also declined significantly after irradiation, and spine morphology changed, shifting from mature mushroom-shaped spines associated with stable, long-term memories toward thinner, more immature forms associated with weaker and less reliable connections. These are not abstract histological findings, they correspond to measurable behavioral changes. Irradiated animals showed impaired spatial memory, impaired recognition memory, impaired episodic memory, reduced executive function, slower fear extinction, and elevated anxiety, a constellation of deficits that maps directly onto the cognitive functions most critical for astronauts, managing complex systems in a high-stakes environment where rescue is not possible and communication, with Earth is delayed by up to 22 minutes each way. And here is the part that should trouble anyone planning a deep space mission. The animals in these studies could not report their own deficits. Their behavior changed, but they continued performing routine tasks. The impairments were most visible in tasks requiring cognitive flexibility, novel problem-solving, and attentional control, exactly the functions a crew would need most during an emergency. An astronaut whose prefrontal cortex has been subtly reorganized by months of galactic cosmic radiation exposure might feel fine, might perform routine checklist operations without difficulty, and might pass a standard cognitive screening while their capacity to respond to an unexpected crisis has quietly degraded. This is the invisible damage at its most dangerous. Not a symptom that triggers a medical alert, but a gradual erosion of the brain's highest-order functions, undetectable from the inside, potentially undetectable from the outside until the moment it matters most. The radiation story converges with the fluid redistribution story in a region of the body no one initially expected: the eyes. Spaceflight-associated Neuro-Ocular syndrome is a condition that was first formally recognized around 2011 under an earlier name, and redesignated in 2017, though the symptoms had been observed for years before either label existed. Approximately 60% of astronauts who have completed long-duration missions aboard the station report some degree of visual change. The clinical findings include flattening of the posterior globe, the back of the eyeball is physically pushed inwards, swelling of the optic disc, where the optic nerve enters the eye, thickening of the retinal nerve fiber layer, choroidal folds, and in some cases a hyperopic shift, meaning the astronaut becomes more farsighted. Some crew members have required corrective lenses in space that they did not need on the ground. In rare cases, the changes have persisted for years after return to Earth. The exact mechanism driving this syndrome remains a subject of active scientific debate, and that uncertainty is itself part of the story. The leading hypothesis involves the cephalad fluid redistribution described earlier. Without gravity pulling fluids downward, the headward fluid migration already discussed persists for the duration of the mission. This redistribution is believed to increase pressure within the skull, intracranial pressure which then transmits along the optic nerve sheath and compresses the back of the eye. The optic nerve, encased in a sheath filled with cerebrospinal fluid, acts as a pressure conduit between the brain and the eye. If the fluid pressure inside the skull rises even modestly, the optic nerve sheath can expand, and the pressure differential across the back of the eyeball changes in ways that physically deform the globe. However, direct measurements of intracranial pressure during spaceflight have been limited, and some parabolic flight experiments measuring pressure during brief periods of weightlessness have found values within normal physiological ranges. This has led researchers to propose alternative or complementary mechanisms. Some suggest that venous congestion in the head, particularly stagnant or slow flow in the jugular veins, which was documented in station crew members, may contribute by raising pressure within the orbital veins and the choroidal blood supply of the eye. Others have pointed to evidence that spaceflight disrupts the glymphatic system, the brain's mechanism for clearing waste products through cerebrospinal fluid circulation during sleep, and that this disruption may alter fluid dynamics around the optic nerve in ways not captured by simple pressure measurements. A 2025 review in Progress in Retinal and Eye Research summarized the current state of understanding by describing the syndrome as likely multifactorial, involving components of fluid redistribution, venous congestion, altered cerebrospinal fluid dynamics, choroidal volume changes, and possibly even individual genetic susceptibility related to 1-carbon metabolic pathways involving folate and vitamin B12. The review noted that sample sizes remain small, only roughly 12 astronauts have been studied in sufficient clinical detail to fully characterize the syndrome, and that the lack of a single well-evidenced etiology reflects the genuine complexity of the underlying physiology. What is not debated is that the structural changes to the eye are real, measurable, and in some cases long-lasting. Optical coherence tomography, a high-resolution imaging technique that produces cross-sectional images of the retina, has documented optic disc edema, increased retinal thickness, and choroidal folds in station crew members. Globe flattening has been confirmed by magnetic resonance imaging. These are not subjective reports of blurry vision. They are objective, instrument-verified, structural deformations of the organ of sight, and the syndrome has been observed exclusively in the context of long-duration microgravity. Short-duration shuttle missions did not produce the same findings. This suggests that cumulative exposure time is critical, that the fluid redistribution must persist for weeks or months before the structural consequences become clinically significant. For a Mars transit crew spending 9 months in microgravity each way, the exposure duration would be far beyond anything currently experienced on the station. Whether the syndrome progresses linearly with time or reaches a plateau remains unknown, because no human has spent long enough in microgravity to answer the question. What this means operationally is easy to overlook, but difficult to overstate. An astronaut whose eyes are progressively reshaping during a mission is an astronaut whose visual capability is drifting away from the corrective lenses that were prescribed before launch. Spaceflight missions carry a supply of adjustable reading glasses in graded diopter strengths for exactly this reason, because NASA already knows that some crew members will need correction they did not need on the ground. But the supply is finite and the prescription changes are not always predictable. A hyperopic shift that exceeds the available lens range leaves the crew member working with compromised near vision for the remainder of the mission. Reading instrument displays, interpreting checklists, examining fine-scale scientific samples, performing medical procedures on a fellow crew member. All of these tasks depend on visual acuity that the syndrome quietly degrades. And the degradation may not be symmetric between the two eyes, which can disrupt depth perception, a faculty that becomes critical during docking maneuvers. extravehicular activity, and any surface operation involving distance judgment on unfamiliar terrain. The astronaut adapts. They squint, hold screens farther away, rely on memory where they once relied on reading. They compensate. But compensation is not correction. It is the brain working harder to extract information from a sensory channel that is delivering less than it used to another invisible tax on cognitive resources that are already under pressure from every other direction. The vision problem is disturbing on its own terms. But it gains additional weight when placed alongside the radiation-driven cognitive changes described earlier, because together they suggest something that no single finding conveys alone. Deep space attacks the central nervous system from multiple directions simultaneously. Radiation degrades neural architecture in the prefrontal cortex and hippocampus. Fluid redistribution deforms the eyes and may alter cerebrospinal fluid dynamics around the brain. The body is not facing one neurological threat. It is facing a convergence of threats that target different structures through different mechanisms, but ultimately degrade the same critical system, the one responsible for vision, cognition, judgment, and decision-making. And the damage at the cellular level is even more pervasive than what shows up in brain scans or eye exams. Every cell in the body contains roughly 6 billion base pairs of DNA organized into 46 chromosomes. This genetic material is under constant assault even on Earth from metabolic byproducts, environmental toxins, ultraviolet light, and natural background radiation. The body repairs tens of thousands of DNA lesions per cell per day through a sophisticated network of repair enzymes. Most of these repairs are accurate. Some introduce small errors. Over a lifetime, these errors accumulate, contributing to aging and increasing the risk of cancer. In deep space, the rate of DNA damage increases substantially, and the quality of the damage changes. Galactic cosmic rays produce clustered lesions, multiple breaks concentrated in a small region of the genome that are qualitatively different from the scattered single-strand breaks caused by terrestrial radiation. These clustered lesions are harder to repair, more likely to result in chromosomal rearrangements, and more likely to produce the kinds of mutations that drive malignant transformation. The body's repair machinery was not calibrated for this type of injury. It attempts repair, but the fidelity of that repair is lower, and the probability of introducing permanent genomic errors is higher. The twins study, NASA's landmark comparison of astronaut Scott Kelly, who spent 340 days aboard the station, with his identical twin brother Mark on the ground, provided direct evidence of this genomic instability. Researchers found that Scott's chromosomes showed increased structural rearrangements, inversions, translocations, and fusions that were not present before flight and were not observed in Mark. His telomeres, the protective caps at the ends of chromosomes that shorten naturally with age, behaved in an unexpected and paradoxical way. They lengthened during spaceflight, possibly due to activation of stem cells, but then shortened rapidly upon return to Earth, ultimately settling at lengths shorter than their pre-flight baseline. Shorter telomeres are associated with accelerated cellular aging, increased cancer risk, and reduced regenerative capacity. But the telomere paradox and the chromosomal rearrangements were not the only lasting changes the study uncovered. Researchers found that a measurable fraction of Scott Kelly's molecular biology had not returned to its pre-flight state 6 months after landing, changes whose full significance would only become clear when scientists examined them in detail. That deeper analysis, and what it reveals about the body's capacity to be durably altered by spaceflight, belongs later in this story. What matters here is the direction of the evidence. Every layer of biological organization the twin study examined: chromosomal structure, telomere dynamics, gene regulation, showed signs of persistent disruption from a single year inside the magnetosphere. A crew in deep space, exposed to the full unshielded flux of galactic cosmic rays for 2 or 3 years, would experience genomic stress significantly beyond what the twin study captured. The cellular damage would be deeper, more widespread, and harder to reverse. This is where the invisible damage becomes most difficult to confront. You can image a thinning bone on a scan. You can measure a flattened eyeball with optical coherence tomography. You can detect a blood clot in a jugular vein with ultrasound. But you cannot feel a chromosomal translocation. You cannot sense a methylation change on a gene promoter. You cannot detect the moment a cell's DNA repair machinery introduces a mutation that will not manifest as cancer for another 15 years. The deepest layer of the damage is written in a language the body cannot read in real time, and by the time it produces visible consequences, the window for intervention may have closed. The radiation does not care whether the spacecraft is functioning perfectly. It does not care whether the crew is healthy, well-fed, well-rested, and psychologically stable. It passes through the hull. It passes through the water supply and the food stores and the exercise equipment. It passes through the skull and into the brain. It threads through the genome of every dividing cell in the gut, the bone marrow, and the immune system. And it does this every second of every day for the entire duration of the mission, accumulating damage that is silent, molecular, and compounding. Everything described so far: the skeletal erosion, the muscular wasting, the fluid redistribution, the cardiac remodeling, the venous stagnation, the radiation-driven DNA damage, the neural rewiring, and the structural deformation of the eyes represents the body under assault from two environmental forces: the absence of gravity and the presence of radiation. But these forces do not operate in isolation. They converge on shared biological systems in ways that amplify each other's effects. And nowhere is that convergence more consequential than in the systems the body relies on to defend itself. The blood that carries oxygen, the immune cells that fight infection, and the cognitive machinery that holds the entire mission together. That convergence is where the damage becomes not merely additive, but something worse. There is a type of damage that does not appear on any scan, does not register on any dosimeter, and does not show up in the mirror. It happens inside the bloodstream, inside the marrow, inside the signaling networks that coordinate the body's defense against infection and disease. It is the kind of damage that a crew member would never notice during a mission, and that a flight surgeon monitoring telemetry from Earth would have no way to see in real time. And yet, it may represent one of the most operationally dangerous consequences of deep space exposure, because it degrades the body's ability to protect itself precisely when that protection matters most. Start with the blood. In 2022, a team led by Guy Trudel at the Ottawa Hospital Research Institute published a study in Nature Medicine that settled a question space medicine had been debating for decades. Astronauts returning from spaceflight had long been observed to be mildly anemic. Their red blood cell counts were lower than pre-flight baselines. For years, the leading explanation was a passive process. When fluids shift toward the head in microgravity, and the body reduces its total plasma volume to compensate, some red blood cells become surplus and are destroyed as a simple byproduct of the volume adjustment. The assumption was that this destruction occurred early in the mission, that the body reached a new equilibrium, and that the anemia resolved relatively quickly after return to gravity. Trudel's study demolished that assumption by measuring alveolar carbon monoxide, a direct byproduct of hemoglobin degradation that serves as a precise marker of red blood cell destruction. The researchers found that hemolysis was not a one-time adjustment. It was persistent. Throughout the entire 6-month missions of 14 astronauts aboard the International Space Station, their bodies were destroying red blood cells at a rate approximately 54% higher than the normal terrestrial rate. Every second of every day in microgravity, the blood was being broken down faster than it should have been. The body compensated by increasing production reticulocyte counts, which measure newly minted red blood cells being released from the bone marrow, were elevated throughout the mission. But the compensation was incomplete. The net result was a state of chronic hemolytic anemia that persisted for the duration of spaceflight. What stunned the researchers was what happened after landing. 1 year after return to Earth, the astronauts' hemolysis rates were still 30% above pre-flight levels. Their reticulocyte counts were still 16% higher. Their hemoglobin concentrations were 3.5% elevated, the body still producing extra red blood cells to keep pace with ongoing excess destruction. A full 12 months after the mission ended, the erythrocytic system had not simply bounced back, it had been persistently altered. The implications for deep space missions are significant. On the station, the anemia is manageable because missions are relatively short and the crew returns to full medical support. But a Mars transit crew would spend up to 9 months in microgravity each way, with the hemolytic process running continuously. The bone marrow, already under stress from radiation exposure and microgravity-induced changes to the marrow microenvironment, would be forced to sustain an elevated rate of red blood cell production for years. And the marrow is not an infinitely renewable resource. The blood-forming stem cells that generate red blood cells, white blood cells, and platelets are among the most radiation-sensitive cells in the body. Galactic cosmic radiation damages these stem cells directly, reducing their capacity to regenerate the very blood supply that microgravity is simultaneously destroying at an accelerated rate. This creates a collision between two independent stressors, microgravity driving hemolysis upward and radiation degrading the marrow's ability to compensate. On a short station mission, neither stressor alone reaches a critical threshold. On a multi-year deep space mission, the two converge on the same biological system from different directions. The body needs more blood cells and simultaneously has less capacity to produce them. The margin of safety narrows with each passing month. And it is not only the red blood cells that suffer. The calcium released from dissolving bones described earlier must be filtered by the kidneys, and the iron released from destroyed red blood cells must be processed by the liver and recycled or stored. Both of these metabolic burdens accumulate. Excess circulating iron is biologically reactive. It generates free radicals through a process called Fenton chemistry, contributing to oxidative stress that damages cell membranes, proteins, and DNA across multiple organ systems. The body is not just losing blood cells, it is being poisoned by the biochemical debris of their destruction. The immune system sits at the intersection of nearly every stressor deep space inflicts on the body. It depends on the bone marrow for its supply of new cells. It depends on the blood for transport. It depends on intact DNA for the genetic instructions that guide immune cell specialization. It depends on the hormonal and neurological systems for regulation. Every single one of these dependencies is compromised in the deep space environment, and the cumulative result is an immune system that is quietly but measurably failing. The evidence for immune dysfunction in spaceflight is extensive and has been building for decades. Studies of astronauts returning from station missions consistently show suppressed T-cell function, the population of white blood cells most responsible for targeting virus-infected cells and coordinating the adaptive immune response. T-cells collected from astronauts after long-duration missions show reduced proliferative capacity, meaning they are less able to multiply in response to a threat. Their cytokine profiles shift, the chemical signals they release to orchestrate immune responses become skewed toward patterns associated with chronic inflammation rather than acute pathogen defense. Natural killer cells, the frontline immune cells responsible for identifying and destroying virus-infected cells, and early-stage tumor cells without prior sensitization, show altered activity during and after spaceflight. Some studies report decreased cytotoxic function, meaning the cells are less effective at killing their targets. This is particularly concerning in the context of cancer surveillance. The body relies on natural killer cells to detect and eliminate the small number of cells that undergo malignant transformation every day. A reduction in natural killer cell function does not cause immediate illness, but it widens the window through which a nascent tumor could escape immune detection and establish itself. One of the most striking demonstrations of immune compromise in spaceflight is the reactivation of latent viruses. Most adults carry dormant herpes viruses, Epstein-Barr virus, varicella-zoster virus, cytomegalovirus, that were acquired during childhood or adolescence and suppressed by the immune system into permanent latency. Under normal conditions, these viruses remain dormant for life. The immune system keeps them contained without effort or awareness. But studies of station crew members have found that the majority of astronauts show reactivation of one or more latent herpes viruses during spaceflight. Viral DNA appears in saliva, urine, and blood samples at levels indicating active viral replication. In most cases, the reactivation is subclinical. The astronaut does not develop overt symptoms, but the fact that these viruses escape immune containment at all is a direct measure of how much surveillance capacity the immune system has lost. On Earth, herpes virus reactivation occurs under conditions of severe physiological stress. Organ transplant patients on immunosuppressive drugs, AIDS patients with collapsed T-cell counts, elderly individuals with age-related immune decline, The fact that otherwise young, healthy, rigorously screened astronauts experience the same phenomenon during spaceflight tells you something about the magnitude of the immune suppression involved. And while the reactivation has been subclinical in most documented cases aboard the station, meaning the virus produced detectable DNA in body fluids but did not progress to overt disease, that distinction offers less reassurance than it appears to. Consider varicella zoster virus. Nearly every adult alive today was infected with this virus. In childhood, it causes chickenpox. After the initial infection resolves, the virus retreats into nerve ganglia along the spinal cord and cranial nerves, where the immune system keeps it dormant for life. When the immune system weakens sufficiently through age, disease, or immunosuppressive treatment, the virus can reactivate along a single nerve pathway, producing the condition known as shingles, a band of painful, blistering lesions that follows the distribution of the affected nerve. On Earth, shingles is painful and debilitating but treatable. Antiviral medications administered early can shorten the course and reduce complications. Most patients recover fully within a few weeks, but shingles has a more dangerous variant. When varicella zoster reactivates in the trigeminal nerve, the major nerve-supplying sensation to the face, it can involve the ophthalmic branch, producing a condition called herpes zoster ophthalmicus. This variant causes blistering lesions around the eye and can directly infect the cornea, the iris, and the retina. Without prompt and aggressive treatment, it can cause permanent vision loss. On Earth, this is managed with intravenous antivirals and close ophthalmological monitoring. In deep space, an astronaut whose immune system has been suppressed by radiation, microgravity-driven cortisol elevation, disrupted sleep, and chronic stress, and whose eyes are already structurally compromised by spaceflight-associated neuro-ocular syndrome, would face a reactivation event in the worst possible context. The onboard pharmacy would carry oral antivirals, but intravenous formulations may not be available. Ophthalmological imaging would be limited, and the underlying immune suppression driving the reactivation would not resolve, because the environmental stressors causing it cannot be removed during the mission. Even when shingles does not involve the eye, it carries a secondary risk that is relevant to crew performance: postherpetic neuralgia. In approximately 10 to 15% of shingles cases on Earth, and in a higher percentage among immunocompromised patients, the pain persists long after the blisters heal, sometimes for months or years. This chronic nerve pain results from damage the virus inflicts on the nerve fibers themselves during reactivation. It can be severe enough to disrupt sleep, impair concentration, and reduce functional capacity. For an astronaut managing complex spacecraft systems during a deep space transit, chronic nerve pain that resists the limited analgesics available on board would represent a direct threat to mission performance, not because the astronaut is dying, but because they are distracted, exhausted, and in pain in an environment where sustained attention is a survival requirement. The varicella zoster scenario is illustrative, not unique. Epstein-Barr virus reactivation, which is the most commonly documented herpes virus reactivation in spaceflight, is associated on Earth with chronic fatigue, lymphoproliferative disorders, and in severely immunocompromised patients certain types of lymphoma. Cytomegalovirus reactivation can cause retinitis, colitis, and pneumonitis in patients with weakened immune systems. These severe outcomes are rare even among immunocompromised populations on Earth, and no astronaut to date has developed them during spaceflight. But no astronaut to date has spent 2.5 years in deep space, with cumulative radiation exposure approaching career limits and immune suppression deepening with each passing month. The margin between subclinical reactivation and clinical disease is a function of immune competence, and immune competence is precisely what deep space erodes. The reactivation data from the station should be read not as evidence that the problem is manageable, but as a warning signal from the body's own surveillance system. Latent viruses do not reactivate because they decide to. They reactivate because the immune system's hold on them weakens. Every reactivation event detected in an astronaut's saliva or blood is a molecular distress flare, a signal that the immune system has dropped below the threshold required to maintain viral containment. On a 6-month station mission, the signal appears and the crew returns before it escalates. On a 3-year deep space mission, the signal would appear and the immune suppression would continue deepening for years afterward, with no possibility of restoring the terrestrial immune environment that kept those viruses dormant in the first place. And the mechanisms driving this suppression are not singular, they are layered. Microgravity alters the physical environment inside immune cells, changing how they sense signals and respond to activation cues. Elevated cortisol from the chronic physiological stress of spaceflight directly suppresses lymphocyte proliferation. Radiation damages the bone marrow stem cells that produce new immune cells, reducing the replacement rate for cells that are lost to normal turnover or killed by radiation itself. The disrupted sleep architecture caused by non-24-hour light-dark cycles aboard the spacecraft interferes with the circadian regulation of immune function, a system that on Earth synchronizes immune cell trafficking and cytokine release to the day-night cycle. Altered nutrition, reduced caloric absorption, and changes in the gut microbiome itself, sensitive to both radiation and microgravity, further degrade the mucosal immune defenses that form the body's first barrier against ingested pathogens. That final point, the gut microbiome, deserves far more than a passing mention, because it connects the immune story to the cognitive story through a biological pathway most people have never heard of, and it represents one of the most recently recognized dimensions of spaceflight damage. The human gut contains roughly 38 trillion microorganisms, bacteria, fungi, archaea, and viruses, collectively known as the gut microbiome. This is not a passive colony of hitchhikers. It is a metabolically active organ equivalent that performs functions the human body cannot perform on its own. Gut bacteria synthesize essential vitamins, including vitamin K and several B vitamins. They break down dietary fibers into short-chain fatty acids that nourish the cells lining the colon. They regulate the maturation and calibration of the immune system from infancy onward. They produce neurotransmitter precursors, including precursors to serotonin and a neurotransmitter, abbreviated GABA, that travel from the gut to the brain through the vagus nerve and the bloodstream, directly influencing mood, anxiety, and cognitive function through what neuroscientists call the gut-brain axis. On Earth, this microbial community maintains a relatively stable composition shaped by diet, environment, sleep patterns, and immune interactions. In spaceflight, that stability breaks down. Studies of astronauts aboard the International Space Station have documented significant shifts in gut microbiome composition during long-duration missions. Microbial diversity decreases the number of distinct species present in the gut drops, and the community becomes dominated by fewer types. The balance between major bacterial groups shifts. Some species associated with anti-inflammatory signaling decline, while species associated with pro-inflammatory activity increase. These changes are consistent with the broader inflammatory shift observed in spaceflight immune profiles, and they may in fact be driving part of that shift because the gut microbiome is one of the primary regulators of systemic inflammation. The mechanisms behind this disruption are multiple and overlapping. The altered diet aboard a spacecraft, heavily processed, limited in fresh produce, lower in fiber diversity than a typical terrestrial diet, directly changes which bacterial species thrive and which decline. Disrupted circadian rhythms affect the timing of gut motility and digestive enzyme secretion, altering the chemical environment the bacteria inhabit. Elevated stress hormones change the pH and mucus composition of the gut lining, favoring certain microbial populations over others. And radiation, particularly the galactic cosmic radiation, that increases dramatically beyond the magnetosphere, damages the rapidly dividing cells of the intestinal epithelium, the single-cell thick barrier that separates the gut contents from the bloodstream. When that barrier is compromised, bacterial products can leak into the blood in small quantities, a condition called increased intestinal permeability, triggering low-grade systemic immune activation that adds another layer of chronic inflammation to an obtuse, already stressed system. The gut-brain axis implications are particularly concerning for long-duration missions. If the gut microbiome shifts away from species that produce serotonin and GABA precursors, the brain's supply of these neurotransmitters may decrease. Serotonin regulates mood, appetite, and sleep. GABA regulates anxiety and neural excitability, A reduction in either compound produced not by direct brain damage but by changes in a microbial community thousands of neural connections away could contribute to the sleep disruption, mood instability, and cognitive changes described elsewhere in this story. The brain is being affected not only by radiation passing through it and fluid pressing against it, but by a chemical supply line that is being disrupted at its source, in the gut by an entirely different set of stressors. This is the kind of damage that no single medical specialty would catch on its own. A gastroenterologist might notice the microbiome shift. A psychiatrist might notice the mood change. A neurologist might notice the cognitive slowing. But the connection between them running through the vagus nerve and the bloodstream, mediated by bacterial metabolites produced in the intestine, would be invisible to any specialist, examining only their own domain. The body is a single interconnected system, and deep space attacks it as such. The gut microbiome is not a footnote to the immune story. It is a supply line under siege, a metabolic and neurochemical infrastructure that sustains both immune function and cognitive function, and that degrades under the combined pressure of radiation, altered nutrition, disrupted sleep, and chronic stress in ways that amplify every other form of invisible damage the body is already accumulating. Each of these mechanisms alone might be compensable. Together, they produce a state of compounding immunodeficiency that worsens with mission duration. On a 6-month station mission, the immune changes are measurable but clinically manageable. The crew returns to Earth, the stresses resolve, and the immune system gradually recovers, though some studies suggest that certain parameters remain altered for months after landing. On a multi-year deep space mission, the stresses do not resolve, they accumulate. The immune system never gets a recovery period. It is suppressed continuously, degraded continuously, and asked to function in an environment where the radiation is simultaneously increasing the mutation rate in every cell it is supposed to be surveilling. The operational risk this creates is not hypothetical. A crew on a Mars transit cannot be evacuated. They cannot receive fresh supplies of medication beyond what was loaded at launch. If a crew member develops a serious infection, bacterial, viral, or fungal, the onboard pharmacy and the crew's own immune defenses are the only resources available. An immune system operating at reduced capacity in an environment where latent viruses are already reactivating, and where the body's ability to generate new immune cells is compromised by radiation, represents a medical fragility that no amount of mission planning can fully mitigate. And the immune story connects directly to the cancer story. The DNA damage from galactic cosmic radiation described earlier does not automatically produce cancer. Most radiation-induced mutations are repaired, and most cells carrying unrepaired mutations are identified and destroyed by the immune system before they can proliferate. Cancer emerges when a cell accumulates enough mutations to escape normal growth controls and when the immune system fails to catch it. Deep space increases the mutation rate through radiation and simultaneously decreases the immune system's capacity to detect and eliminate mutant cells. These two trends, rising mutagenesis and falling immune surveillance, move in opposite directions along the same timeline. The longer the mission, the wider the gap between the threat and the defense. This is not a risk that manifests during the mission. It is a risk that the crew carries home with them, a legacy written into their genomes and their immune memory that may not produce clinical consequences for years or decades. An astronaut who completes a 3-year Mars mission and returns in apparent good health may be carrying an elevated cancer risk that will follow them for the rest of their life. The invisible damage does not end when the mission does. It echoes. Now add the brain. The cognitive effects of deep space exposure are not a separate category of damage. They are the downstream consequence of everything already described: radiation remodeling neural circuits, fluid redistribution altering intracranial dynamics, disrupted sleep degrading memory consolidation, chronic inflammation reaching the central nervous system, immune-mediated neuroinflammation, adding another layer of insult. The brain does not exist in isolation from the body. It is perfused by the same blood that is being hemolyzed. It is bathed in the same cerebrospinal fluid whose dynamics are altered by microgravity. It is irradiated by the same galactic cosmic rays that are damaging the marrow and the gut. Every systemic stressor described in this story converges on the organ responsible for keeping the crew alive. The animal data on radiation-induced cognitive impairment, discussed earlier, provides the most direct evidence, but the human data, while more limited, is consistent with the pattern. Astronauts returning from long-duration station missions report subjective cognitive changes, difficulty concentrating, slower recall, increased mental effort for tasks that were previously automatic. Standardized neurocognitive testing administered before, during, and after station missions has detected subtle but measurable changes in processing speed, working memory, and attentional performance. These deficits are small in absolute terms and generally resolve within months of return. But they are real, they are consistent, and they occur after missions of only 6 to 12 months in an environment where radiation exposure is a fraction of what deep space would deliver. The psychological dimension cannot be separated from the physiological one, and it is a mistake to treat it as a softer, less serious category of risk. Psychological strain in deep space is not merely emotional discomfort, it is a neurobiological state that compounds every other form of damage. Confinement in a small spacecraft with a fixed crew for years, with no possibility of leaving, no possibility of rescue, and no possibility of real-time communication with Earth, creates a chronic stress environment that has been studied extensively in analog settings. Antarctic winter-over crews at isolated stations like Concordia, submarine deployments lasting months, and NASA's own ground-based analogs, including the Human Exploration Research Analog and the Mars 500 isolation study, which confined 6 volunteers in a sealed habitat for 520 days. The findings from these analogs are remarkably consistent. Prolonged isolation and confinement produce measurable cognitive decline that worsens over the second and third quarters of the confinement period. Crew members show decreased vigilance, increased lapses in attention, reduced motivation for non-essential tasks, and subtle impairments in interpersonal communication that can escalate into conflict. Sleep quality degrades progressively. Even when sleep duration is maintained, the architecture of sleep shifts, with less time spent in the deep slow-wave stages most critical for memory consolidation and neural repair. Hormonal profiles change. Cortisol rhythms flatten, losing the sharp morning peak and evening trough that characterize healthy circadian function. This flattening has been associated on Earth with chronic fatigue, depression, and impaired immune regulation. The Mars 500 study, conducted from 2010 to 2011 by the European Space Agency and the Russian Institute for Biomedical Problems, found that crew members became progressively more sedentary and more prone to sleep disruption as the mission progressed. One crew member spent most of the latter portion of the study in a state researchers described as behaviorally consistent with chronic sleep deprivation, sluggish, cognitively slowed, and socially withdrawn even though his total sleep hours were nominally adequate. The quality of engagement degraded even when the quantity of rest did not. What makes these analog findings so relevant to deep space is that they occurred without any radiation exposure, without microgravity, without hemolysis, without immune suppression, and without the bone and muscle loss that adds physical discomfort and fragility to the picture. The psychological degradation documented in these studies represents a baseline, the minimum cognitive cost of confinement and isolation alone. A real deep space mission layers every physiological stressor described in this story on top of that baseline. Consider the compounding arithmetic. A crew member 6 months into a Mars transit has been losing bone density since departure. Their muscles have atrophied despite daily exercise. Their blood is being destroyed faster than normal, and their marrow is struggling to keep pace under radiation bombardment. Their immune system is suppressed, with latent viruses reactivating in their saliva. Their eyes may be changing shape as fluid accumulates behind the retinas. Their prefrontal cortex has been absorbing heavy ion tracks that are subtly reorganizing its neurotransmitter networks. Their sleep architecture is degraded by a light-dark cycle that does not match any natural rhythm. Their cortisol regulation has flattened, and they are confined in a volume smaller than a studio apartment with the same 3 or 4 people they have seen every day for half a year, with a 20-minute communication delay making real-time conversation with anyone on Earth impossible. No single one of these factors, taken in isolation, would necessarily incapacitate a well-trained astronaut. But they do not occur in isolation. They occur simultaneously, and they interact. Sleep disruption worsens immune function. Immune dysfunction promotes neuroinflammation. Neuroinflammation degrades cognition. Cognitive degradation reduces the quality of decision-making around exercise, nutrition, and sleep hygiene. Poor sleep hygiene worsens sleep disruption. The loop feeds itself. Radiation-induced changes to prefrontal dopamine signaling reduce attentional control. Reduced attentional control makes it harder to sustain the 2-hour daily exercise protocol that partially protects bone and muscle. Reduced exercise accelerates musculoskeletal decline, which increases physical discomfort, which disrupts sleep, which worsens cortisol dysregulation, which further suppresses immune function. Every system's failure mode becomes an input to another system's failure mode. This is what it means to say that deep space attacks the body as an interconnected whole. The danger is not any single effect. The danger is the convergence, the way that bone loss and anemia and immune suppression and radiation damage and cognitive decline and psychological strain all share biological pathways, all worsen each other, and all progress along timelines that are individually manageable but collectively unprecedented. And there is a feature of this convergence that makes it uniquely treacherous. The person experiencing it may be the last to know. Cognitive impairment has a well-documented feature in both clinical neurology and aerospace medicine. It degrades the capacity for self-assessment. A person with impaired executive function is, by definition, less able to evaluate the quality of their own executive function. This is not a theoretical concern. It has been observed in sleep deprivation studies, where subjects who have been awake for 24 hours consistently rate their own performance as adequate, while objective measures show significant decline. It has been observed in altitude sickness, where climbers above 8,000 meters make decisions they would recognize as irrational at sea level, but cannot evaluate correctly at altitude. And it has been observed in confinement studies, where crew members who showed the most pronounced cognitive decline on standardized tests were often the same crew members who reported feeling fine. On the station, this problem is mitigated by constant communication with mission control, routine psychological screening, and the knowledge that the crew can be brought home within hours if necessary. In deep space, none of these safeguards exist in their current form. Communication delays of up to 22 minutes each way make real-time psychological support impossible. The crew must function as an autonomous unit, making critical decisions about vehicle operations, medical treatment, and mission priorities without immediate ground oversight. If the crew's collective judgment has been subtly eroded by months of compounding physiological and psychological stress, there is no external system to catch the error. The Mars 500 study offered a preview of this dynamic. Despite being conducted on the ground with no physiological stressors beyond confinement, the study documented instances where crew members made decisions that, in retrospect, reflected impaired judgment errors of omission, failures to follow established protocols, and interpersonal conflicts that escalated beyond what the crew's own conflict resolution training could manage. The researchers noted that these failures were not dramatic. They were quiet. They accumulated. They represented a gradual erosion of group function that was difficult to identify from inside the system. Now imagine that same dynamic playing out inside a spacecraft, where the crew is also anemic, assignment suppressed, radiation exposed, sleep deprived, and operating with prefrontal cortices that have been physically remodeled by heavy ion bombardment. The margin for error does not just narrow, it narrows at the same time the capacity to detect and correct errors is being reduced. This is the deepest operational challenge of long-duration deep space missions, and it is almost entirely invisible. There is no alarm that sounds when cognitive reserve drops below a threshold. There is no biomarker that lights up red when the collective judgment of a crew has degraded past the point of reliable self-monitoring. The damage is distributed across too many systems, mediated by too many interacting mechanisms, and masked by the human brain's remarkable ability to construct a feeling of normalcy, even as the substrate generating that feeling is quietly deteriorating. Every system described across this story so far, bones, muscles, cardiovascular, eyes, blood, marrow, immune cells, neurons, neurotransmitters, sleep architecture, hormonal regulation, and psychological resilience is under simultaneous assault from the moment a crew leaves Earth's magnetic and gravitational protection. The damage is cumulative, the interactions are multiplicative, the timeline is relentless, and the body's own reporting systems, pain, fatigue, discomfort, the subjective sense that something is wrong are among the least reliable indicators of how far the degradation has actually progressed. The question that remains is not whether deep space damages the human body. That is settled. The question is whether the damage can be stopped, slowed, or reversed well enough to keep a crew functional across a mission lasting years rather than months, and whether the body that returns to Earth after such a mission would still be recognizable as the body that left. That question does not have a comfortable answer. In April of 2019, the results of NASA's twin study were published in the journal Science. It was the most comprehensive molecular portrait of a human being before, during, and after spaceflight ever assembled. 10 research teams across the country had spent years analysing samples from astronaut Scott Kelly, who spent 340 days aboard the International Space Station, and his genetically identical twin brother, Mark, who remained on the ground. The dataset spanned genomics, epigenomics, transcriptomics, proteomics, metabolomics, immune profiling, cognitive testing, cardiovascular imaging, and telomere biology. It was, as one of the lead researchers described it, the most detailed view ever obtained of the response of the human body to spaceflight. The headline finding was cautiously optimistic. The majority of the physiological changes Scott experienced in orbit, shifts in gene expression, metabolic alterations, immune recalibration, cardiovascular remodeling returned to baseline within 6 months of his return to Earth. The body had been stressed, but it appeared to recover. The system bent, but it did not break. But buried inside that reassuring summary was a quieter finding that carried far more weight for the future of deep space exploration. 7% of Scott Kelly's gene expression changes did not return to pre-flight levels 6 months after landing. These were not random genes. They included genes involved in DNA repair, the molecular machinery the body uses to fix radiation damage and copying errors in the genome. They included genes governing immune function, the regulatory circuits that control how the body detects and responds to infection and malignancy. And they included genes related to bone formation, the signaling pathways that direct the skeleton to rebuild itself under mechanical load. 7% sounds small. It is not. The human genome contains roughly 20,000 protein-coding genes. 7% represents approximately 1,400 genes whose expression was still altered half a year after the mission ended. 1,400 instructions being read differently. 1,400 molecular switches that had been flipped by spaceflight and had not flipped back. This is what it means to say the body was rewritten, not metaphorically, not poetically, literally. The environment of spaceflight, the combination of microgravity, radiation, fluid redistribution, immune stress, disrupted sleep, and confinement had changed how Scott Kelly's cells read their own DNA. The genetic code itself was not altered. The sequence of bases remained the same. But the epigenetic layer, the system of chemical tags and structural modifications that determines which genes are active and which are silent in any given cell, had been durably changed in ways that persisted long after the prodem. Environmental trigger was removed, and Scott Kelly spent his mission inside the magnetosphere. He was partially shielded from galactic cosmic radiation. He was resupplied every few months. He could communicate with the ground in near real time. His mission lasted under a year. Everything about his experience represented the most protected, most supported, most moderate version of human spaceflight currently possible. A crew bound for Mars would have none of those protections. The minimum energy transit to Mars takes approximately 7 to 9 months each way, depending on orbital alignment. The surface stay, dictated by planetary mechanics, lasts roughly 500 days. The crew must wait for Earth and Mars to realign before the return window opens. Total mission duration from launch to splashdown, approximately 900 days, 2.5 years, and for the transit portions, roughly 600 days combined. The crew would be in deep space, beyond the magnetosphere, exposed to the full unshielded flux of galactic cosmic rays inside a spacecraft that cannot be meaningfully resupplied or reached by rescue vehicles. Every stressor described in this story would be operating for the entire duration of that mission, and not one of them levels off. Bone loss does not plateau at a safe threshold, it continues for as long as gravitational loading is absent. Hemolysis does not stabilize, it persists at elevated rates throughout microgravity exposure. Radiation damage does not accumulate to a ceiling, and stop each day adds new DNA lesions to a genome that is simultaneously losing repair capacity. Immune suppression does not reach a floor, and hold it deepens as the bone marrow degrades, as sleep architecture deteriorates, and as chronic cortisol exposure continues to blunt lymphocyte function. Cognitive erosion does not announce itself and wait for correction. It compounds silently behind a wall of subjective normalcy that becomes less reliable the further it progresses. The compounding math is what transforms a collection of individually manageable risks into something that may be collectively unsurvivable without intervention. So the obvious question becomes, what interventions exist? The honest answer is that countermeasures for deep space physiological damage are in various stages of development, and none of them are ready. For bone and muscle loss, the most effective current countermeasure is resistive exercise, the 2-hour daily protocol, using the advanced resistive exercise device aboard the station. This approach significantly reduces musculoskeletal decline compared to earlier eras of spaceflight, but it does not eliminate it. Astronauts still return from 6-month missions with measurable bone deficits, and the protocol demands a substantial portion of the crew's waking hours' time that would be even more precious on a Mars mission with a heavier operational workload. Pharmaceutical countermeasures have been explored, primarily bisphosphonates, a class of drugs used on Earth to treat osteoporosis by inhibiting osteoclast activity and slowing bone resorption. Some studies have shown promise in reducing spaceflight-related bone loss when combined with exercise, but the drugs carry side effects including gastrointestinal irritation and, with long-term use, a paradoxical risk of atypical bone fractures. Prescribing a medication for 2.5 years to a crew with no access to advanced medical care if complications arise introduces its own risk calculus. For radiation, the options are more limited and more sobering. Passive shielding, the physical material between the crew and the radiation source, is effective against solar particle events, which consist mostly of protons that can be stopped by sufficient mass. Aluminum, polyethylene, and water all provide meaningful protection against solar storms. But galactic cosmic rays are a different problem entirely. The heavy ions that constitute the most biologically damaging component of galactic cosmic radiation are so energetic that stopping them with passive shielding requires impractical amounts of mass. Worse, when a heavy ion strikes a shielding material, it can undergo nuclear fragmentation, producing a shower of secondary particles, lighter ions, neutrons, gamma rays that may actually increase the radiation dose to the crew inside the spacecraft. Thicker shielding against galactic cosmic rays can, counterintuitively, make the problem worse depending on the material and the particle energies involved. Active shielding, using magnetic or electrostatic fields to deflect charged particles before they reach the spacecraft, has been proposed and studied for decades. The concept is sound in principle. If Earth's magnetosphere can deflect galactic cosmic rays, perhaps an artificial magnetic field could do the same for a spacecraft. In practice, the engineering challenges are formidable. Generating a magnetic field strong enough to meaningfully deflect the highest energy galactic cosmic ray particles would require superconducting magnets of enormous scale and power, operating continuously for years in the deep space environment. No such system has been built or tested at the required scale. Current designs remain conceptual, and the mass, power, and reliability requirements place them well beyond present engineering capability. Pharmacological radioprotection drugs that reduce the biological impact of radiation after it has entered the body represents another avenue of research. Some compounds under investigation aim to scavenge the free radicals produced by ionizing radiation before they can damage DNA. Others attempt to enhance the body's own DNA repair mechanisms. Animal studies have shown modest protective effects for certain agents, but translating these results to humans is complicated by the need for chronic administration over years, the potential for side effects that could impair crew performance, and the fundamental difficulty of protecting against the clustered DNA damage caused by heavy ions, which is qualitatively different from the diffuse damage these drugs were designed to address. For the immune system, no spaceflight-specific countermeasure currently exists. Researchers have discussed the possibility of immune-boosting therapies, targeted cytokine supplements, periodic vaccination boosters to maintain immune memory, or even prophylactic antiviral medications to suppress herpes virus reactivation. But none of these approaches has been validated for long-duration deep space use, and the interactions between immune-modulating drugs and the other physiological stresses of spaceflight are poorly understood. An intervention that boosts one arm of the immune system might suppress another. In the complex, multi-stressor environment of deep space, predicting the net effect of any pharmacological intervention becomes extraordinarily difficult. The most promising single countermeasure, the one that would address the widest range of physiological damage simultaneously, is artificial gravity. If a spacecraft could generate a continuous gravitational field equivalent to Earth's surface gravity, or even a significant fraction of it, many of the microgravity-driven problems would be reduced or eliminated. Bone loss, muscle atrophy, fluid redistribution, cardiac remodeling, and possibly even some components of spaceflight-associated neuro-ocular syndrome are driven primarily by the absence of gravitational loading. Restore that loading, and the body's structural maintenance systems would continue operating as designed. The engineering concept most frequently discussed is a rotating spacecraft, or a rotating habitat module. By spinning a structure at the appropriate radius and angular velocity, centripetal acceleration can simulate gravity along the outer wall. The crew would live and work on the inside of the outer ring, with their feet pointing outward and their heads pointing toward the rotation axis. The concept has been explored in engineering studies since the 1950s and is familiar from science fiction. The spinning station in 2001, A Space Odyssey, is the most iconic depiction, but building such a system for a real Mars mission involves challenges that remain unsolved. The radius of rotation matters enormously. A small radius centrifuge spinning quickly enough to generate 1 g of acceleration at the rim would produce a steep gravity gradient across the height of a standing human, significantly stronger at the feet than at the head, and would generate Coriolis forces that disrupt balance and spatial orientation. To minimize these effects, the radius needs to be large, ideally 100 meters or more. A rotating structure of that scale, built robustly enough to support a crew for 2.5 years, represents a spacecraft far larger and more massive than anything humanity has launched. The engineering is not impossible in principle, but it would require a development program of a scale and cost that no space agency has yet committed to. Smaller radius centrifuges, short-arm centrifuges, that a crew member lies on for a few hours per day are a more near-term possibility and are under active study. The idea is to provide intermittent gravitational loading sufficient to slow bone and muscle loss without requiring the entire spacecraft to rotate. Results from ground-based analogues are mixed. Some studies show benefits for bone density and cardiovascular function. Others find that the intermittent exposure is insufficient to fully counteract the effects of the remaining 20 or more hours per day spent in microgravity. The optimal dose, duration, and frequency of centrifuge exposure remain unknown. And a short-arm centrifuge does nothing to address the radiation problem, the immune problem, or the cognitive problem. This is the uncomfortable reality that defines the current state of deep space human spaceflight. For every major category of physiological damage, musculoskeletal, cardiovascular, hematological, immunological, neurological, and genomic countermeasures either do not exist only in early research stages, or exist in forms that are partially effective but insufficient for missions lasting years rather than months. No single intervention addresses the full spectrum of damage. No combination of currently available interventions has been demonstrated to maintain human health across a mission of Mars-class duration. And the interactions between different countermeasures, drugs that might conflict with each other, exercise protocols that compete for crew time, shielding materials that produce secondary radiation, add layers of complexity that have barely begun to be explored. The gap between where the countermeasures are and where they need to be is not a gap of willingness or imagination. It is a gap of biology. The human body evolved over hundreds of millions of years inside a very specific set of environmental parameters, a 1 g gravitational field, a magnetic shield generated by a molten iron core, an atmosphere that filters ultraviolet and absorbs cosmic radiation, a 24-hour light-dark cycle driven by planetary rotation, and a biosphere that provides nutritional complexity no spacecraft pantry can replicate. These are not optional features. They are the operating conditions the body requires to maintain itself. Remove them, and the body does not simply become uncomfortable. It begins to disassemble itself according to rules that made perfect biological sense on Earth and make no sense at all in deep space. Bones dissolve because the body has no reason to maintain mass it is not using. Muscles atrophy because metabolic resources are redirected away from tissue that serves no function in weightlessness. Blood cells are destroyed at accelerated rates because the cardiovascular system is recalibrating for a fluid distribution that has fundamentally changed. The immune system downregulates because the hormonal and circadian signals that sustain it are disrupted. DNA. Repair fidelity decreases because the genome is absorbing damage from particle types it was never exposed to across the entire history of terrestrial life. None of this is malfunction. All of it is the body doing exactly what it was designed to do, adapting to its environment. The problem is that the environment is one the body was never designed to adapt to. The adaptations that serve the crew in microgravity reduced bone mass, reduced muscle mass, reduced blood volume, reduced cardiac output are precisely the adaptations that will endanger them the moment they step onto a planetary surface or return to Earth. The body optimizes for the present at the expense of the future, because evolution never anticipated that the present and the future would require such radically different configurations. This is the deepest and most unsettling truth about what deep space does to the human body. It does not merely damage you. It recruits your own biology as the instrument of that damage. Your cells are not failing, they are responding. Your bones are not breaking, they are remodeling. Your immune system is not collapsing, it is adapting. Every change documented across this story, from the thinning femur to the flattened eyeball, to the reactivated virus to the shortened telomere, represents the body doing its job in an environment where doing its job means slowly dismantling the systems it would need to survive anywhere else. And the changes are quiet. That word has appeared throughout this story because it is the single most important characteristic of the damage. Quiet. Not dramatic. Not painful. Not alarming. The astronaut floats through the spacecraft, performs experiments, exercises for 2 hours, eats a rehydrated meal, sends a message home, and sleeps. Everything feels manageable. Everything appears normal. The mission proceeds according to plan. And beneath that surface of routine competence, 1,400 genes are being read differently. Bone microarchitecture is being irreversibly remodeled. Neural circuits in the prefrontal cortex are being reorganized by particle tracks that pass through without sensation. Red blood cells are being destroyed 54% faster than they should be. Latent viruses are waking up in the salivary glands. Natural killer cells are losing their ability to identify the tumor cell that just divided in the lining of the colon. The body does not send a notification for any of this. There is no equivalent of a warning light on a cockpit panel. The damage accumulates below the threshold of perception, below the resolution of the crew's own self-assessment, and in many cases below the detection capability of the onboard medical equipment. It exists in the space between feeling fine and being fine, a space that, on a short mission, remains narrow enough to cross safely, but that on a multi-year deep space mission may widen into a gap the crew cannot see and cannot bridge. Artemis II flew its crew around the moon and back in 10 days. They returned safely. The radiation exposure was brief. The microgravity duration was short. The physiological impact, by all accounts, was minimal. But that mission was not a rehearsal for Mars. It was a proof of concept for the vehicle, not for the body. The spacecraft performed. The question of whether human biology can perform across a mission 50 to 100 times longer in a radiation environment, orders of magnitude more intense, remains not only unanswered but, in critical respects, untestable with current infrastructure. You cannot know what a 3-year deep space mission does to a human body without sending a human body on a 3-year deep space mission. And you cannot ethically send a crew on that mission without reasonable confidence that they will return in a condition that allows them to live the rest of their lives without being defined by the damage the mission inflicted. This is the paradox that sits at the center of deep space human exploration. The only way to fully characterize the risk is to take the risk. The only way to validate the countermeasures is to test them in the environment they are designed to protect against. And the environment itself may be changing the test subjects in ways that compromise their ability to report whether the countermeasures are working. The script that Hollywood writes about space danger will always favor the explosion, the breach, the fire, the villain. These are stories that have beginnings, middles, and ends. They have moments of crisis and moments of resolution. They are narratives the human mind can process because they follow the architecture of threat and response that our brains evolved to understand. The real danger of deep space does not follow that architecture. It has no crisis point. It has no single moment where everything changes. It is a slow curve bending in the wrong direction across every system in the body simultaneously. Too gradual to trigger alarm, too distributed to localize, too quiet to feel. It is the skeleton thinning at a rate the hands cannot detect. It is the immune system losing surveillance capacity, while the genome accumulates mutations it can no longer catch. It is the prefrontal cortex reorganizing its dopamine networks while the person it belongs to believes their judgment is unimpaired. It is the blood being destroyed and rebuilt and destroyed again in a cycle the marrow may not be able to sustain for the years the mission requires. The body that boards the spacecraft at launch and the body that returns to Earth 2.5 years later may look the same from the outside. The astronaut may walk down the steps, wave to the cameras, and smile for the press conference. But at the molecular level, at the cellular level, at the level of gene expression and immune memory and bone microarchitecture and neural connectivity, it may no longer be the same body. Deep space does not kill you all at once. It rewrites you, quietly, progressively, system by system, in a language you cannot read and a silence you cannot hear. The question humanity faces is not whether we can build a spacecraft capable of reaching Mars. We probably can. It is not whether we can engineer life support systems that keep a crew breathing and fed for 3 years. We are close. It is not whether we can navigate the orbital mechanics or land on the surface or launch again for the return trip. These are hard engineering problems, but they are engineering problems, and engineering problems yield to engineering solutions. The question is whether the human body, this specific biological system, shaped by this specific planet, calibrated to this specific gravity, and this specific radiation environment and this specific magnetic shield, can be transported across interplanetary distances and returned in a condition that we would recognize as healthy. Whether the astronaut who steps onto Mars after 9 months of transit is still capable of the physical and cognitive performance the surface mission demands, whether the astronaut who boards the return vehicle after 500 days on a planet with 38% gravity and minimal magnetic protection still has the bone density to withstand re-entry forces, the immune function to survive a post-landing infection, the cognitive clarity to manage a spacecraft through the most dangerous phase of the mission. The answer may be yes with countermeasures we have not yet developed, with technologies we have not yet built, with medical interventions we have not yet validated. The human capacity for solving problems that once seemed impossible is real and should not be underestimated. But the answer today, in this moment, with the tools currently available, is that we do not know. And the reason we do not know is that the damage is invisible. It hides inside the body, beneath the skin, below the threshold of sensation, behind the reassuring competence of a well-trained crew performing well-rehearsed tasks in a well-designed spacecraft. It hides, and it accumulates, and it waits. The deepest challenge of deep space is not the void between planets. It is the quiet catastrophe unfolding inside the only vessel that actually matters, the one made not of titanium and carbon fiber, but of calcium and collagen, hemoglobin and neurons, DNA and memory. The spacecraft can be rebuilt. The body may not offer the same option, and that is the invisible damage. Not a single wound, but 1,000 silent changes, converging across years, rewriting the body from the inside, in the dark, without pain, without warning, without permission. The most dangerous thing about deep space is not what it does to you. It is that you might never feel it happening until the day you need the body you used to have and discover it is no longer there.